Biology by Tiffany Taylor. How does cloning work? Cloning means to make an identical copy of. Replicate a fragment of DNA placed in an organism so that there is enough to analyze or use in protein production. Dolly and Bonnie. Dolly was the first mammal with an adult cell to be successfully cloned. Dolly was cloned, lived and died at the Roslin Institute in Midlothian, Scotland. Cloning animals. Obtaining eggs from a cow is slightly easier. That's why cows have been cloned more than other animals. In China, a commercial animal cloning center is currently being built to help meet the country's rapidly rising beef demands. How cloning started. In 1880, an animal was cloned for the first time. German biologist Hans
Drish, I think. I don't know.
Cloned a sea urchin. How a clone is different from an identical twin. The clone is artificially made in a petri plant, unlike in the case of twins, wherein the fertilization is done internally in the mother's womb. Twins result from the natural splitting of one zygote or the fertilization of two ova.
Yeah.
While clones are a result of the harvesting of a foreign somatic cell or foreign ovum from a donor. George W. Bush. He set limits on government-funded stem cell research. The cellular building blocks that developed into different parts of the body. Bush called for passage of a Senate bill sponsored by Senator Sam Brownback that would make it a crime to carry out human cloning. Clones. Scientists routinely produced cells of all kinds of human and animal cells for research purposes. The issue in the Brownback bill is whether to make it a crime to do experiments in which a copy of somebody's DNA is inserted into a human egg. Human cloning. Taking someone's DNA like a skin cell or hair follicle and inserting it into an unfertilized human egg from which the existing DNA has been removed. Because the human egg grows, divides, and gives rise to all the cells of the body, researchers think they will be able to use the genetically altered egg to produce a lab dish full of certain kinds of human cells, such as neurons and
pancreatic cells, I think.
Pancreatic cells. Aren't researchers more interested in cloning for its therapeutic benefits? Those who oppose cloning on moral grounds believe that these embryos are human beings and therefore experimenting on them, even at the earliest stages of development, is wrong. Many consider this research to be ethical and very promising. Benefits of therapeutic cloning. If you have a condition resulting in the loss of brain cells, then implanting new brain cells might correct or ease the symptoms. But such implants could be rejected by the body if they came from another donor, in the same way that liver or kidney transplants are often rejected. With therapeutic cloning, doctors could theoretically take a sample of their DNA, put it into a human egg, and grow neurons genetically identical and thus unlikely to be rejected.